Hi guys! So I know there's not going to be a lot of viewers for this one, but because it's AHL Bio. Okay, so today we're going to be talking about photosynthesis, but like the really complicated parts. So you should probably watch the first video that I made about 2.9 about photosynthesis because that'll make it way easier for you to understand this one. So first we're going to jump right in and start with the two types of reactions. We have our light dependent and light independent and light dependent. Yes. So the products of light independent, well, let's start with the products of light dependent. We have O2, we have ATP, and we have NADPH. So in light independent, we have O2 and we have glucose. So we're going to start off with light dependent. So I'm going to draw a little doodle here. So we're going to have this electron transport chain. If you remember from the Calvin cycle, we're going to start with something called a photosystem. And we're going to have another photosystem here. And then we're going to have our ATP synthase over here. I'm just going to draw some primary things. Okay. I'll tell you what these are all in a second. Okay, so we're going to start off over here with photosystem 2. PS means photosystem 2. So in photosystem 2, we have something in the middle called a reaction center. And this is going to be chlorophyll A. Chlorophyll A. So this is what we're going to call our reaction center. Basically, around this reaction center, we have a lot of pigments. These green things are all pigments. And in the middle, we have an electron. There's going to be an electron over here. So when the light from the sun comes down on the leaf, the light will go and be absorbed by all the different pigments, which in this case are chlorophylls. However, there's also going to be some xanthophylls and carotenoids, but it's going through these chlorophylls, and this energy is all going to go towards the reaction center and the electron in the reaction center. This electron is going to get so excited that it's going to jump. Oh, let me also explain further. Over here, we're going to be in the stroma, which is the inside of the chloroplast. And over here, we're going to be in the thylakoid space. So we're going to be in the thylakoid space over here. So this electron is going to be hopping from over here. It's going to hop into the proteins. In the stroma, we have some protons just floating around because it's the stroma. There's everything in there. So these protons are going to go and pass through this protein using active transport because they need energy from the electron. Active transport, and they're going to be transported into the thylakoid space. Now, this electron is going to keep hopping, but let's talk about what happens in the thylakoid space for a second. So these protons are being pumped by using this electron. However, the electron is gone from chlorophyll A in the reaction center of photosystem 2. Now, how are we going to replace this electron? You might be thinking, or you might not. But if you are, and you have to anyways, we need to go back to something called the photolysis of water. So the photolysis of water is the splitting of water. That's what it literally means. It's the splitting of water into an electron, a proton, and oxygen. Oxygen is going to leave. Let's put an arrow leaving the note because we don't need it. It's a waste product. This proton, you see all the protons. It's going to be pumped with all the protons. And the electron, let's do this in yellow. Electron is going to make its way and take the place of the electron that was gone from the reaction center. Okay, now that we're done with that, we're going to move on to photosystem 2. I have to write that here. Photosystem 1. It's kind of funky because photosystem 2 comes before photosystem 1, which is contrary to chronological order. So in photosystem 1, we also have a lot of pigments. And the job of photosystem 1 is basically to reduce the electron it's to reduce NADP plus into DPH. So that is what's happening in photosystem 2. It's a reduction of NADP to NADPH. And that's done by one of the enzymes, ferredoxin, I think. Yes. You can just look it up in case. So we're going to think of ferredoxin over here. It's a protein, does this job. We don't have to know it though. So the electron's going to keep hopping. It's going to power all these things to happen. And then it gets to ATP synthase. This is a lot of people's favorite enzyme. I don't have a favorite enzyme just yet. So it's going to hop to ATP synthase and in here. So now going back to these protons that are just floating around the thylakoid space. Something about the thylakoid space is that it's a very tight, it's very narrow. That's why it's very, very easy to build a concentration gradient. Now, after this concentration gradient starts building and they start overdoing and we have too many protons, they're going to go back through the ATP synthase and back into the stroma. However, as the protons go through, we're going to pretend that this proton is just moving along its way. The ATP synthase is going to add a phosphate onto some things inside ATP synthase, and we're going to produce ATP. Isn't that crazy? I don't know where all this stuff came from, but we just know that it's going to add some phosphates to some things inside ATP synthase, and it's going to make ATP. And that's how you get ATP. So let's name some of these processes now. I'm sorry, this is a very complicated drawing, but we should be able to understand it after we know the processes now. All right, so in this process, we're going to have chemiosmosis. This is a valuable thing to know. Chemiosmosis is the diffusion of protons through ATP synthase. Well, it's a transport of protons through a semi-permeable membrane, I believe, through to produce a concentration gradient and stuff like that. And another thing we have to know is phosphorylation, because we're phosphorylating the protons or whatever is moving through ATP by using light, in the presence of light. All right, perfect. Now, moving on. So what did we collect? Never mind, not moving on. What did we collect from this? We're going to do our products in red. We got ATP, that is very dark red. We got ATP. We also got O2. And we know where these were used. And that's it, right? Those are both of our things. ATP and O2. Oh, and we got NADPH over here. So all of them have been completed. And that is it for light-dependent photosynthesis. Now, moving to light-independent. Here's where we get to the Calvin cycle. So we're going to be starting off with a 5-carbon thing. 1, 2, 3, 4, 5. So this thing has 5 carbons. It's 5C, let me call it a molecule. It's a 5C molecule. Okay, so what we're going to do in the Calvin cycle is we're going to add this, and it's called RUBP. I believe that stands for something like ribose biphosphate. And that is the sound of rain outside. And we're going to add it to a carbon. And this carbon is going to be coming from, let's not do that. It comes from CO2. So we're using CO2. And what do we know about CO2? It is in the environment. So it's in the environment and we're taking it to use in the Calvin cycle. That's why plants uptake CO2 and they give out oxygen. So we're using 1C. So we're going to add those together basically. And we're going to get a 6-carbon molecule. That's quite a molecule. And we don't need to know what it's called. We just need to know that it's called, that it has 6 carbons. So 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5. Okay, 6 carbons. Now it's going to go through lysis, and it's going to break into 2 pairs of 3. 1, 2, 3, 1, 2, 3. These 2 are called G3P. Now, this glycerate phosphate, or something like that, glycerate triphosphate, maybe, it's going to be reduced. Usually we reduce other things and oxidize the carbons. But in this Calvin cycle, we're going to be reducing these, which is kind of strange. But NADP. Oh, this is the NADPH from before. Remember when we made NADPH here? It's coming into handy. Good job. So it turns into NADP plus. And we're going to use another one here, which is going to go from NADPH to NADP plus. Plus. And to do that, we're also going to be using an ATP turning to ADP. Okay, so we're using some ATP here. I'm not no quantities, quantity X, okay? Yes, so we're going to do that. And from there, we're going to get 2 triose phosphates, actually. That is hideous. 1, 2, 3, 1, 2, 3. And these are going to be the reduced versions, and that's why they're so high in energy, because they have so many electrons. They're stealing the electrons from NADPH. So these are very, very, very high in energy. Okay. So we're going to have a C. These are called TP, triose phosphates. Now, here comes some math parts. Over here, we started with 6 RUBPs, and we added 6 CO2s. That's why in the equation, it's 6 CO2. Very, very simple to remember. So we have 6 Cs. Don't get confused with the numbers here. And that's why, okay, so if we're doing some math here, we're adding 6 of 5. So we have 30 Cs here, because we have 5 carbons, so which is 30 carbons. And here we have 6 1 carbons, so we have 6 carbons. And we add those, we get 36 carbons. So going into this whole process over here, we have 36 carbons. Here, no carbons are added or lost. They're just being changed a little bit. So at the end of this, we also have 36 carbons. However, here's the thing. Listen closely. These 36 carbons, 6 of them, 1, 2, 3, 4, 5, 6, are going to come together and make a 6C, which is eventually going to turn into glucose. Glucose, yes. Oh, it's going to turn into glucose. Okay, so those are the 6 C's. They're going to leave, and they're not going to be part of this Calvin cycle anymore because we already made our glucose, which is our byproduct. Can we check that off the list now? Oh, we already made our oxygen. Now we finished making our glucose. So amazing. Now we're going to keep going with the cycle though. We're not done yet, guys. We still have more. So these, okay, let's do more math. So at the end of this, we got 36 C's. I'm gonna put this 36 C's inside so we don't get confused in that section. Now, after we took these 6 C's away, so we're gonna have 36 C's minus 6 C's, that's what to make glucose. We're gonna have 30 C's again. Oh, look at that. In this cyclic metabolic pathway, to get to 30 C's again, we have accomplished it. Tick. Okay, beautiful. Now, over here, we just have some rearrangement. That's not spelled right. Rearrangement. And just like refixing all of the things to extract all the possible energies we can extract. So, basically, we are done. And we have completed the Calvin cycle successfully. I think that deserves a round of applause. And obviously here we're using some energy, so our ATP is being converted into ADP, but that's fine because we're making so much ATP in our chain here. So it funds for our endeavors, basically. And that's it, guys. That's the Calvin cycle. Thanks for watching. If you have any questions, let me know. Bye.